Good afternoon, everyone. So as people I'm going to start. It is noon. We're just going to go ahead and power through as people continue to join us. I am so excited to introduce my dear colleague and friend of many, many, many years. Oh my goodness. Dr. Margaret Sheridan. She is an assistant professor of psychology and neuroscience at UNC Chapel Hill. She received her PhD from the University of California, Berkeley, go Bears, in clinical psychology. Dr. Sheridan uses multi-method tools, including fMRI, EEG, and psychophysiology to examine how early experiences shape the developing brain, leading to risks for psychopathology. Her research ambitiously examines these factors in cross-sectional, longitudinal, and experimental designs. She focuses in particular on the period of early childhood and adolescence, two periods of maximal change in the neural architecture of the prefrontal cortex. Dr. Sheridan was a Robert Wood Johnson Health and Society Scholar from 2007 to 2010 at the Harvard School of Public Health and Harvard Medical School. Her research is funded by several grants, including the National Institutes of Mental Health, the National Institute of Child Development, and the National Science Foundation. She is the co-PI of the Network on Reversibility within the National Institutes on Aging and a board member for the Flux Society for Developmental Cognitive Neuroscience. Dr. Sheridan has published extensively with 90 publications and 8 book chapters. And are you still rock climbing too? Rearing 2 children, managing the household.
Yeah. Mostly traded the children for the rock climbing. So.
Yeah, I hear you. Well, it is a great pleasure, Margaret, to have you here. Please join me in welcoming Dr. Sheridan today. Take it away.
All right. It is so wonderful to be here. I first just want to say thank you so much, Barbara, for the invitation, and thank you all for coming. It's so exciting to even virtually be back in the Bay Area. I have to say it was maybe my favorite part of this was I had a bunch of colleagues from my Berkeley days saying, hey, I saw that you were on the schedule. And I thought, oh man, I wish I was going to be there to give everybody a hug. So today I'm going to talk about the dimensional model of adversity and psychopathology. And the question that I'm going to answer, which is the central question that I ask in my research in the at UNC in the Circle Lab, is how is brain development linked with adversity, and how does this contribute to risk for psychopathology? And childhood adversity is associated with long-term deficits in health and well-being very broadly across many forms of outcomes, many forms of health problems, many different outcomes, and also very broadly with regards to how adversity, different kinds of adversities, which can predict those outcomes. And this has been described as social environments and experiences getting under the skin in life in ways that affect the course of human development. And given how much research has been done examining the association between adversity experienced in childhood and deficits in health and well-being, that's the fact of that association is really not going to be the focus of my talk today. And that's particularly because in addition to those numerous association studies, there's also evidence from randomized control trials that these associations are causal. That as we know that exposure to prolonged adversity in early life leads to changes in the brain, behavior, and physiology. So I think this is no longer a question. But what is worth asking about, and what I'm going to spend my time talking today about, is how adversity gets under the skin. What are the mechanisms which link these exposures with those health outcomes later? And I think this is a particularly important question to be asking because it can tell us a little bit about how to intervene appropriately with different populations with the different kinds of adversity exposure. So as a clinical psychologist, that's sort of important to me. And I want to talk, the most common approach right now is the ACEs approach. And this is an approach which takes a number of different kinds of adversities, sums them up, and looks at the association between the number of adversities that somebody's been exposed to and the kinds of outcomes and any particular outcome. So here we're looking at the number of comorbid outcomes. You can look at the number of, you can look at the presence of any given disorder. You can look at the number of given disorders that somebody's diagnosed with. You can look at comorbidity between physical and mental health outcomes. There's sort of any number of things when you put on the y-axis, and you'll see this kind of linear or curvilinear association between adversity and those sorts of outcomes. But before I get too much into that approach, I want to talk a little bit about what we're talking about when we talk about adversity. And I'd like you to think about these two kids. So the kid on the left, we can call him John. I think it's your left. He has a father who has a problem with alcoholism and is given to intermittent explosive rage, in which he harms both John and also his mother. So he's sort of witnessing a fair amount of violence in the home, and he's experiencing some of that violence himself. But he also has a very invested caregiver in his mother, who spends a lot of time working with him in developmentally appropriate ways and scaffolding him in a lot of different forms of his development. In contrast, we have a kid like Sarah. Sarah has a single parent. That parent has to work multiple jobs in order to make ends meet, and Sarah is unable to have a lot of scaffolded interactions for her development with her caregiver or any other invested caregiver. So she's really sort of alone a lot of the time. In the ACEs approach, these two exposures would be considered the same. So you would just add that if you had both of those exposures, you'd have a 2. If you had multiple other kinds of exposures like divorce or poverty, you'd get higher scores. And this is a dominant approach. As I said, there's been lots of good research linking these two things. And the mechanism of this model, the proposed mechanism, is a stress mechanism. So the idea is that you are exposed to lots of different kinds of adversity. Each of those forms of adversity increases your stress physiology a little bit, so results in a stress reaction. And that increase of stress leads to these future outcomes. And that mechanism this model is a really excellent way of identifying who's going to have a disorder. So if you're a clinician and you're sitting in your office and you're wondering, to whom do I allocate the resources that I have? Which person here is most likely to have a problem later on, given just their adversity exposure? The ACEs model is a great model for you to be using because it will tell you who is likely to have a problem. But I think that this particular mechanism is really limited. And the primary way I think it's limited is that I'm thinking about child development. And I think this mechanism is fundamentally a decay mechanism. The idea and the way this has been mostly studied in animal models is that you have a functioning system. That system is buffeted by stress exposures, and the system's ability to recalibrate in response to those stress exposures eventually declines, and it becomes unable to handle the stress that it's experiencing. But children's brains aren't developed systems, and we know that they're highly plastic and designed to take input from the environment to shape the way they function in the future. So thinking about the classic neurodevelopmental mechanisms that we know shape brain development, not thinking about those when thinking about adversity, I think is a real miss. So we know that the brain develops dramatically, both structurally and functionally, from early childhood into adulthood. And this is just one picture of cortical thinning across age in a longitudinal study, sort of showing that really dramatic development. And we know that one of the reasons why you see these structural changes that we're seeing here, one of several reasons, is there are actual differences in the presence of synapses and the number of dendritic spines in the cortex, so that you see this proliferation of synapses early in development, and then subsequent pruning and refining of the circuits that are established really early on. I mean, you see this here in a postmortem study looking at increases in synapses across early childhood, decreases across childhood, and then eventually sort of a plateau in adulthood. So in my work, what I've thought about is what are those actual experiences that kids are having that might be shaping their brain development using these developmental plasticity mechanisms. And the two kinds of exposures that I and my colleague, Kate McLaughlin, came up with when we were developing this model were deprivation and threat. And the idea here is that threat would be an exposure, would be a dimension of experience. And there would be a number of different exposures that are named exposures, which would contribute to the threat experience. So something like physical or sexual abuse would be an exposure to threat, domestic violence, and community violence. And something like poverty really sits right in the middle of this diagram because it puts you at risk for threat and deprivation, but it doesn't actually mean that you're going to be exposed to threat and deprivation. So plenty of children in poverty spend their time over here in the sort of typical development cohort or quadrant. In contrast to something like threat, deprivation is really characterized by the lack of input. So here we're thinking of things like institutionalization, which would be a really robust lack of input where the child would have very little input from any invested caregiver, through something like neglect, where those inputs would be lacking, but they would be intermittently present. And then something like the kind of low resource environments which result in people not being able to invest fully in their children that are associated with poverty would be another example of sort of places where you might imagine there'd be deprivation exposures, and you could go in and measure those in the home. And in thinking about these two dimensions, we were really imagining them as ways of characterizing what the proximal inputs into the child's brain are. So as opposed to the sort of intention of the parent, we're actually not thinking about what the parent wanted to do, but instead what actually happened to the child's brain and how that might shape brain development. So the hypotheses we have here are that different dimensions are going to affect the brain and behavior differently, and there'll be robust controls for the other dimension. So that what we expect threat to do is to shape the parts of the brain that are involved in threat processing, and we expect deprivation to function really differently. We think that the impact of adversity on the brain here would be a pathway to psychopathology. So we want to be able to show that ultimately, not only do you see these different dimensions affecting brain development differently, but also that that ultimately leads to differences in psychopathology. And finally, in a new part of the research that we're doing now, we're really trying to show that dimensions are natural kinds, so they can emerge from data-driven approaches. And this is sort of a project I'll talk about a little bit at the end. Now, before we get into the meat of the talk, I want to talk about the principles and limitations of this model. So importantly, I don't think that these two dimensions of adversity describe everything in all of childhood adversity. So for example, I think there are, and I don't think they describe all the different mechanisms through which adversity comes to shape brain development. Even there are other forms of exposures to adversity, something like unpredictability, a real variability in the caregiving environments that is difficult to predict from the perspective of the child, might affect brain development in a different way through the same developmental plasticity mechanisms. In addition, I think, as I described earlier, I want to be clear that we're really describing the proximal inputs into the child's developing brain. So sometimes, and we can talk a lot about this later if anyone's interested, when I describe something like deprivation or threat, people sort of immediately go to the attributions of the caregiver and what they sort of are trying to do or not trying to do, and how sort of warmly they're regarding their child. I think it's important to remember that sometimes parents who regard their child very warmly, in some instances, behave very differently with them in other instances. And also some parents who regard their child very warmly are just unable to provide their child the inputs that they would want to do, given the other circumstances of their lives. So we're really not talking about the intention of the caregiver, just what the brain is experiencing. And then finally, we expect these proximal inputs to lead to proximal outcomes. So while I was describing that ACEs is a good way to predict health, I think that this DMAP model might be a better way to predict how you get to those health outcomes. So this is sort of the proximal input to the brain, changing the way that the brain functions, and then that ultimately leading to health outcomes. Okay, so I'm going to talk a little bit about what each of these dimensions are and what we're expecting to have happen as a result of them, and then some research we have so far supporting these ideas. So threat exposure, the concept here is that threat exposure are a set of early learning experiences that are enriched for violence, and that they're going to enhance neural systems which support dealing with danger. This idea of threat exposure initially came out of some work in rodents that we were reading from Regina Sullivan, and she's got a bunch of different work in her lab looking at this. But the sum of most of this work shows this particular pattern, which is that if you take pups and you look at their ability to do learning very early in infancy, all pups will do this attachment learning where they'll form an affiliative sort of an approach bond with any stimulus, noxious or otherwise. And then later in infancy, they do that same kind of attachment learning if their mom is present. But late in infancy, they have sort of fear learning, which we see usually in humans, which is that they avoid noxious stimuli and they approach positive stimuli. Interestingly, if you take those same pups, or pups just like them, and you rear them either with what's called an abusive mother here, which is sort of a dam who's been stressed out so that she behaves in abusive ways with her pups, or if you just go ahead and just straight up shock them when they're infants, little pup rats, what you see is that early in infancy, they still show this attachment learning, but later in infancy, they begin to show fear aversion. So earlier in their development, they're able to do this appropriate fear learning where they're avoiding noxious stimuli. And interestingly, this trauma exposure in really early life in the infants is associated with a bunch of outcomes in rodents that we sort of think are related to negative outcomes, health outcomes in humans. So we hypothesized, based in this literature and a rich literature in humans looking at things like exposure to maltreatment, that what we would see is that threat exposure would specifically influence the development of neural systems involved in fear learning. So that specific thing that we saw there in the rodents, and also emotional reactivity. And we expected this to include the amygdala and the medial prefrontal cortex. And the general idea here is that you could take a bunch of different kinds of exposures to threat. I'm showing you here different ways you could measure that. These would all affect the system, and then that would be the mediator to outcomes like externalizing and internalizing psychopathology. And initial studies, we sort of came up with this idea and wrote some papers about it. And we also happened to be running a study of maltreatment at the time. And so we were able to test some of these initial ideas, partially test them in this existing sample, which we called the stress and adolescence study. And in this sample, we had 50 participants that were adolescents. About 38% of them had abuse exposure by self-report. And so that is they reported being exposed to something that would meet their criteria for abuse. And about 21% had parents with less than a high school education. We did some neuroimaging with them while they were doing this emotion regulation task, which is a really commonly used task. And the thing I want to just bring our attention to is that here they were looking at a neutral or a negative image. And sometimes when they were seeing that negative image, they were asked to decrease their response to the negative image. So we sort of taught them cognitive reframing techniques and asked them to use those while they were looking at the negative image. Whoops. Oh, it just disappears. Okay, well, I'll just tell you what we found. So during that, when we were asking them to decrease their response to that negative image, if we compared that to just looking at a negative image, what we saw is increased amygdala activation for adolescents who'd been exposed to abuse relative to those who hadn't been exposed to abuse. So basically their amygdala remained active even despite using these kinds of regulation techniques. Now, I wanted to repeat this work in early childhood. And so I'm going to describe a couple of different studies where we're able to get a little closer to the hypotheses that come straight from the rodent literature, because we're able to look earlier in childhood. A lot of the work that we've done before was all in adolescence. Now, this is some work I'm going to describe now, which was done by Laura Macklin, a graduate student in my lab who is fantastic. And in this study, she ran 64 kids between the ages of 4 and 7, so much younger, and we were able to intentionally measure deprivation and threat. So here we have, for deprivation, the child's own report of neglect, some information about the cognitive scaffolding in the home, and then parental education. And for threat, we have the child's own report of exposure to violence, the parents' report of partner violence, so domestic violence, and then abuse predilection, which is a questionnaire that sort of assesses how likely it is that parents are going to be involved in abuse behavior. All of our analyses here are going to control for age, gender, and IQ. And importantly, depth and threat in the sample was uncorrelated. That varies across samples, and I can talk a lot about that later if people are interested. So here we did a fear learning study. This is getting really close to what we saw in those rodent studies, trying to really replicate that kind of work. What we did here is we showed children in between 4 and 7 a blue square, and nothing happened. Then we showed them an orange diamond, and some of the time, about 80% of the time, a noise would happen with the orange diamond. And then we showed them that same orange diamond, but no noise. And then we did blocks of this and sort of repeated it a bunch of different times. And this block design fear study is something that's been done previously with little kids. It speeds up the fear learning process so that you can get more trials in without keeping the kids sitting there for a long time. We looked at skin conductance response. Specifically, we looked at it in 2 places. We looked at it here in blocks where they were hearing that negative noise the kids didn't like to hear. And we also looked at it here when they were seeing the orange triangle, but there was no longer a negative noise. And these are 2 different kinds of things. In the first case where the noise is on, that's their sort of body's ability to mount a response to the presence of a noxious stimuli, period, what that looks like. And in the second case, it's their body's ability to predict the fact that a noxious stimuli is going to happen, given the fact that this cue is coming on that's associated with it. And that second thing is called fear learning. That's the real fear learning response. This first thing is sort of response to a negative stimulus. And we'd seen in other studies looking at, and many other people had seen in other studies looking at kids exposed to maltreatment and other forms of violence, a blunted response, a blunted physiological stress response to this kind of negative stimulus. And that's also what we saw here. So if you plot the response to the negative stimuli and by threat exposure, what you see is a decreased response to the negative stimulus, the more threat exposure that you have. And this is controlling for deprivation. We also saw this interaction with age, and this is driven by just a few kids. And so we're replicating this now in a much larger sample, and we'll see whether or not it replicates. But in this small sample, what we see is that kids who are younger and exposed to threat acquire this fear learning. So they have a larger response to the orange diamond, even when there's no sound around. They have a larger skin conductance response to that than kids who are low threat. And also by the time everybody gets sort of a past 6, they all are able to acquire this differential response to this, the orange diamond. So they're able to show fear learning. So we are here kind of replicating this thing that we'd seen previously in rodents, which is that kids with maltreatment exposure earlier do in fact show an earlier fear learning than kids without that. Now, that's not the specific hypothesis we had. Of course, our initial hypotheses were all about the brain. And so we also wanted to see whether or not we were seeing the same differential association with the brain in early childhood. And we were able to do that in this existing data set of kids between the ages of 4 and 11. About 68% of them have usable MRI data, and half of them have anxiety. So this is actually an anxiety study that happened to collect a lot of data about exposure to maltreatment and other forms of threat. And we were interested both in the effect of threat and deprivation on brain structure, and also if that interacted with their risk for anxiety. We're also following up this sample now with a much larger study where we're able to follow the original 917 kids, and they're now all in adolescence, and we're able to ask more of those questions. But the spoiler for this is I'm just going to talk about the effect of threat and deprivation on brain structure, because there was no interaction with early risk for anxiety, which was a real surprise to me and contrary to everything that I proposed in the grant. So we'll see what happens with the larger sample. And I would be very interested in hearing people's thoughts about that if people have some later. So here we're going to measure deprivation using neglect and a self-report of parental involvement in terms of sort of reading books at home and reading books before bed. For threat, we again have domestic violence, so abuse, and also a sort of indicators of a violent environment, again by parent report. There's also here a cumulative risk variable, which is a bunch of other major stressors, things like being in a car accident, having a parent die, having a major illness as a child. Those sorts of things we controlled for in these analyses. So that was sort of an attempt to control for other bad things that happen in childhood that are not specifically deprivation or threat. We used FreeSurfer to analyze structure of the brain. So we're going to look at the amygdala, hippocampus, striatum, and we're separately looking at the caudate, putamen, and accumbens. We're going to look at surface area in the whole brain and thickness in the whole brain. What we see, first of all, is that if you look at the association between threat and cortical structure, you see that kids exposed to threat have both a smaller amygdala and hippocampus, and that's controlling for deprivation, cumulative risk, anxiety, age, gender, and which scanner they were scanned on. And you also see that they have smaller surface area. So while deprivation didn't predict cortical surface area, threat predicted really big differences in surface area in the lateral and also medial prefrontal cortex, and here in sort of extrastriate medial temporal cortex. So across these two studies, and we have observed here that in early childhood and adolescence, threat exposure is linked with increases in emotion reactivity, earlier fear learning, and associated changes in the amygdala and prefrontal function and structure. This then we replicated in a lot of other studies. So we see reduced differentiation between fear and safety cues in adolescents, and we see decreased automatic and explicit emotion regulation in kids with exposure to threat. And in all of these cases, we're defining threat by interpersonal violence in the home and in the community. So what about deprivation? Our hypotheses for deprivation are really different. So instead of thinking about deprivation as the presence of an atypical learning experience, we're thinking of it as a lack of a species typical learning experience. So we think there are cognitive and social inputs that your brain is expecting, and that the richness of those will predict are sort of like a form of cognitive stimulation, both cognitive and social stimulation. This is kind of a graphic of what we're imagining. We think that in early childhood, kids are supposed to be getting these inputs through their interactions with an invested caregiver that would be using, tools like toys and books and their sound of their voice and their hands to interact with the child and kind of help them with their cognitive and social development. That the child's brain in receiving this input, especially in association cortex, is going to increase or keep a lot of the dendritic spines and branches that it has already formed in very early childhood and infancy. Then we'll be able to measure that as cortical thickness later in life. And in contrast, we imagine a situation in which a kid isn't getting those inputs. So there's a lot of species expected inputs that it's not receiving. Then the brain, in order to create the most efficient set of connections that it would want for the future environment that it expects to inhabit, given these early inputs, would prune away a lot of those dendritic spines and branches, and the result would be also decreases in cortical thickness that you could observe later. So here the way I've really thought about this is that these are sort of the expected inputs for association cortex, both prefrontal and parietal cortex. And here I'm showing you sort of lateral prefrontal cortex and the middle frontal gyrus and inferior frontal gyrus. And the idea is that deprivation would shape the development of those areas of association cortex that are involved in higher order cognition. And then we would expect, consequently, that higher order cognition would also be different in kids who are exposed to deprivation. And this is really a theory by analogy. I'm thinking of when we were first developing this theory, we were thinking of examples from rodents and humans where sensory input had been really dramatically decreased or eliminated. And in those cases, what you see are changes in dendritic arborization and cortical thickness in primary sensory cortex. So here we were thinking that childhood adversity would change the kinds of inputs that were the same kind of species expected inputs for association cortex. So the same things that were supposed to be shaping association cortex there are sort of certain kinds of inputs that were expected for sensory cortex, and these were the ones that were expected for association cortex. And the reason why we thought this is because we had some evidence early on that institutionalization was associated with cortical thinning and reduced volume in middle childhood. And more recently, we've seen that their experimental variation exposure to institutionalization early in childhood does change frontal lobe thickness in adolescence. Also, we had then a little bit of evidence, and now we have more, that a lack of cognitive scaffolding in the family was associated with selective deficits in executive function and language. And we can go back to those same exact studies that I was just talking about and ask whether or not we see in those studies where we saw this evidence of threat exposure, do we also see evidence for this other pathway, this deprivation exposure, when we control for maltreatment? So we want to take out the variance associated with that exposure to threat and just look at the variation associated with deprivation. Now, our first study wasn't designed to test that, and so all we have as a deprivation exposure is parental education. I don't think this is the right way to go, but that's what we had at the time. And if you look, I think it is, despite the fact that I think as a variable, parental education isn't the right variable for measuring deprivation, I think it's pretty interesting and sort of, I think, profoundly demonstrates the power of this kind of disassociation, that if you look at parental education, it predicts this very simple working memory task. We're remembering 2 versus 4 items. It predicts your performance on the task, and it also predicts your activation of parietal cortex during the task. And that association is robust to controls for maltreatment. And this is the same sample where we saw that really, we saw that 38% of them had been exposed to maltreatment. It had that effect on amygdala function and emotion task, and also for age and gender. And this is just showing that in the whole brain. You see that in parietal cortex, and you also see it here in the middle frontal gyrus and in the anterior cingulate cortex. In that early childhood study where we looked at the kids doing the fear learning task, we also gave them a cognitive control task. It's a very simple task where you have to press on the same side if something is pink, and if it's yellow, you withhold that pink press, and you withhold that same side press, and you do an opposite side press. So it's a Simon task, and it assesses inhibitory control. And what we see in that same group of kids is that if you look at deprivation, it predicts your performance on that task, and it's robust to controls for threat, age, gender, and IQ. So you see this sort of mirror of the impact of threat. You see the specific impact of deprivation here. And then finally, in the study where we were looking at neural structure related to threat, we also looked at deprivation. And here we see that deprivation did predict cortical thickness, and that threat was not associated with cortical thickness. And again, this is controlling for cumulative risk, anxiety, threat, age, gender, and scanner. Now, the one thing we did see is that deprivation predicted thicker cortex and not thinner cortex. And there's actually a variety of evidence from the literature on sensory deprivation about whether or not you do complete deprivation or partial deprivation, if you should expect thicker or thinner cortex. So I think we're still working out the exact mechanisms down to the level of sort of how the increases and decreases of pruning would be expected early versus late in development. And so it is true that it's not exactly what we predicted, but this is sort of very close to being pretty consistent with the kinds of things we thought would be happening based on our initial work. And so across the studies I've described, I think we have pretty robust evidence, and we're still doing more studies of this, but that the different dimensions of adversity are going to affect brain and behavior differently, and they'll be robust to controls for the other. The other thing that we hypothesize is that the impact of adversity on the brain here would be a pathway to psychopathology. As a clinical psychologist, I think it's sort of interesting if adversity affects the brain and behavior differently, but not as interesting or as important to the outcomes that I care about if it's not a mediator to psychopathology ultimately. And in order to really test this, we've needed some big longitudinal studies with rich measurement of a bunch of different variables, and we've found 2 of them so far. And we've tested what we could test in those 2 big samples. The first one is the Child Development Project. And in this project, this is done with Adam Miller, who was a postdoc in my lab and is now a research assistant professor, and it's also fantastic. In the Child Development Project, this was a sample that was collected by Ken Dodge in the 1990s, so it's from a while ago. There's 585 kids, and about half of them are female. And when those kids were between the ages of 5 and 6, they measured deprivation, or they measured the number of toys, books, child books, whether or not there was an appropriate play area for the kids in the home. So they went into the kids' home and did that. And they also measured the parents' report of abuse between the ages of 0 and 5, harsh discipline, family conflict currently. And then during the home interview, they observed whether or not they saw hostility or rough physical contact between the parent and the kid. When the kids were 14, they came back in and they measured vocabulary. And that's one of the only outcomes in this study which sort of you could neatly put into either the deprivation or the threat bin. So this sort of the mediator that would make sense. So that's the one we were able to measure. And then at age 17, they had internalizing and externalizing on the CBCL. We used a SEM mediation model with bootstrap resampling to test whether or not our entire model of a mediation by deprivation through vocabulary predicted psychopathology later. And what we observed is that it did. We saw that deprivation predicted verbal abilities, and that mediated the association to externalizing psychopathology. And in contrast, threat was unrelated to verbal abilities, but had a direct association with internalizing and externalizing psychopathology. And these associations were robust to controls for socioeconomic status. So here we're able to move away from this idea of socioeconomic status as a proxy for either deprivation or threat. And then we replicated that recently in the Fragile Family study. And I'm going to talk more about this study in a minute. But here we have a larger sample of about 2,300 kids. We have the same longitudinal design. So we're shifting our developmental periods a little earlier, but we have deprivation and threat measured between 1 and 3. Using very similar indicators to what I just described in the child development study, although a much richer set of indicators. Verbal abilities at age 5, using actually very similar measures of verbal ability. So in the child development study, it was the Peabody picture vocabulary. Here it's the Peabody picture vocabulary and one of the subscales from the Woodcock-Johnson. And then at year 15, we have internalizing and externalizing psychopathology. And again, what we see is that deprivation is mediated, the effect of deprivation on both internalizing and externalizing is mediated by verbal abilities, whereas threat has a direct effect on those 2 outcomes. So the first set of hypotheses that we had coming from the DMAP hypothesis, our model, was were pretty well supported by the initial data that we were able to collect or analyze. But one of the questions I had is whether or not these were natural kinds. And I kept wondering whether or not they would emerge from more data-driven approaches, in part because I think in science, as I was saying to my students recently when we were talking about how we do science, I had this idea. I really liked this idea. I felt like it made a lot of sense to me. And I felt like there was ways that I was testing or thinking about the idea that were probably really affected by the degree to which I personally thought it was right. And I wanted to see whether or not if I was let the sort of degrees of freedom vary more dramatically, if I would still see this kind of these dimensions emerge from the data. And so lots of people have thought about this before, in the sense that many people have tried to think about how adversity exposure clusters. And previous work has asked sort of that question using our typical tools in psychology. And those typical tools rely on the co-occurrence or correlation of different kinds of exposures. And if you do that analysis, what you see is something like stuff that occurs with poverty. So those things cluster together. Something like institutionalization is an unusual exposure in the US, and it's kind of off on its own. And there's things that co-occur with abuse. And this is really describing who is likely to have multiple of these things happen to them. But it's not really describing the thing that we're trying to get at in this model, which is that some of these are going to cause certain kinds of outcome or certain kinds of mediators, and other of them are going to cause other kinds of outcomes or mediators. And so I decided to take a network analysis approach, and I thought this would be a good approach because it's a very general solution to examining structure within a set of relationships. It's very agnostic about what kind of data you use. You can put anything into it. And the methods for describing clustering and the role of specific nodes are really robust. And so I thought we could sort of use that work other people had done. And the idea here is that in this network analysis, you would introduce into the network analysis, in addition to these predictors, the outcomes that you thought would be associated with the predictor. So things like cognitive control, cortical thinning, fear learning, emotional control, or amygdala reactivity, for example. And that introducing those variables will sort of reorganize your networks. So you'd see those original associations that you see usually in cluster analyses, but now the presence of these mediators would sort of reorganize the networks into two clusters, so they would sort of pull apart things that were co-occurring by providing them other things to be strongly associated with. And we published one paper on this recently, and I'll just describe briefly the method that we used here. So we started with two different data sets that we had collected. One was about 200 kids and one was about 150 kids. And we ran the network analysis on one, and then we replicated it on the next data set. And it's not like a machine learning thing. It didn't learn anything from doing one to try out on the other. It's just two different analyses of two different data sets using the same method. In both cases, what we did is we took the original data set. There was some data missing in both data sets. So we imputed a bunch of different data sets, 1,000 different data sets. On each of those, we created networks, and then we averaged those together to create an average network. So that of all the times, this sort of imputation is just a way of bootstrapping to create a bunch of different data sets. And then we asked whether or not this average network, we now have the average network, and we have a confidence interval around that network that we've got because we imputed all those data sets. And we can ask whether or not that network, that average network, is similar or different to a proposed cumulative risk model. And the cumulative risk model that we're proposing is there are sort of two different ones. We checked it against both. One would be a cumulative risk model where everything is connected to everything. All the exposures are connected and all the outcomes are connected. And another cumulative risk model would be one where all of the exposures are connected to all of the outcomes. That's really the testing. There's no differential relationship between exposures and outcomes. Then the other thing we did was we took the original data set, we imputed them, created correlation networks, and then on each of those we did a clustering algorithm to see whether we used 4 different clustering algorithms to ask how you would differentiate these clusters of symptoms, or sorry, clusters of predictors and outcomes. And then we did a consensus clustering. And then we asked whether or not that consensus clustering looks similar or different to the deprivation and threat model, which is the hypothesized model that I had in my mind when I was putting these variables into the network. And I did not come up with these network methods myself. There's an excellent scientist, Bill Shee, who was at UNC, who helped me do this work and is an author on the paper. And then he unfortunately went to Amazon to make more money than he can make at UNC. So, but he is still working on this a little bit with us, which is great. So this is an example of one of the first times we did this. This is one of the first 2 studies that was in that initial paper. And what you can see is that this is the average network and with the average clustering indicated on it. And as you can see, we do see that we get one cluster, which is like a threat cluster, and then one cluster, which looks like a deprivation cluster. So over here in the threat cluster, we have performance on the emotional Stroop, the effect of emotion on dot probe, things like sexual abuse, domestic violence, physical abuse, and direct community violence, and the emotional Stroop adaptation score. So things having to do with emotion and exposure to violence. Over here, we have performance on vocabulary, matrix reasoning, and that's clustering with the home environment questionnaire, income to needs, parent education, and 2 forms of parent education. There's a couple of things that didn't work the way we expected. One is that parent reported physical neglect clustered with this sort of threat cluster. So the parent was reporting on the same instrument their physical abuse, the physical abuse the child had been exposed to, and the physical neglect the child had been exposed to, and the sexual abuse, and all of that clustered together. That was also true in the other network that the other network analysis we performed in another data set in the same paper. So in those two analyses, we sort of got closer to what we were expecting to see, but it wasn't exactly the thing we expected. And it's possible that we were wrong in thinking that physical neglect belonged over in the deprivation cluster, but it's also possible that there's just so much shared measurement variance that you can't really pull them apart. And there was a second data set, this Fragile Families data set, where we were able to, I think, ask a little bit more about that. So this Fragile Family data set has, you can select the sample that you're using a number of different ways. Here we're working with 2,500 parent-child dyads, about half of them are female. It was identified, the people participating in the study, it's a representative sample, so they identified as people in urban centers with high rates of single parent families. We're looking here just at their age 3 data following their enrollment at birth. And what's nice about the Fragile Families data set is that it's a big data set. We're able to control for lots of things. That's one of the most exciting things about it. And it's also a very rich data set, so it has lots of different kinds of exposures in it. So in terms of our controls, here we're now able to control for age and gender, as we've been doing, but we're also able to control for prenatal risk. So we have information about the parents, the child's prenatal exposure to alcohol, smoking, and illegal substances. We have information about their birth weight and their prenatal visits. So how many prenatal visits the parents had. We're able here to control for parental education and income to needs, parental race, and parental cognitive function. So the parents' performance on the same cognitive function tasks that we're going to look at in the kids. And then as in all network analyses, we're also controlling for all other variables in the network. So this is our measurement of predictors. So here we have things that we thought were pretty clearly cognitive and social stimulation. Cognitive stimulation measured in the home, types of toys, number of books, time spent outside, positive social stimulation as observed in the home interview. So where the people who are measuring the home kind of go there to the parent's house and measure those things in the home. We had things that were pretty clearly material deprivation, food insecurity, parent report of neglect, and whether or not the people who had gone to the home observed the child appeared neglected. Then we had some things we thought we're pretty confident were threat, emotional abuse, physical violence, community violence, spanking before the age of 1, and domestic violence. And then we had these 4 variables that we weren't quite sure what they would be. So we thought they were sort of represented chaos or unreliable access to primary caregivers. They were frequent separation, eating meals together, difficulties with childcare, and flexible work schedule. So sort of indicators of struggling with specific kinds of resources. And how that affects the family. And then we had some outcomes. So one of our outcomes is the PPVT, the Peabody Picture Vocabulary Test, which showed up in some earlier analyses. It's something people gave kids a lot in these studies, in these large studies. And also we have internalizing symptoms, externalizing symptoms, and positive social behavior as rated by the parent when the kid is 3. So this is probably psychopathology, but it's also sort of some general tendencies. We're just taking dimensional scores here, so nothing more, no cutoffs. And what we see is 2 clusters. So we didn't get 3 clusters, and we didn't get 1 cluster. We got 2 clusters, and it does look like the deprivation cluster does have many of the indicators that we thought would be related to deprivation. So these light blue ones are all of our cognitive stimulation variables, and they are clustered with the PPBT, which is here, the number 1. The threat variables all clustered together, and they all clustered with symptoms of psychopathology or and poor social skills. But also in the threat cluster, we see that self-reported neglect is also, and food insecurity are both clustered with threat. So these are these 2 variables right here. But interestingly, observed neglect, so this is the parent's report of neglect, is clustered with the parent's report of maltreatment or abuse, but the observed neglect, whether or not the person going to the home thought the child looked neglected, is now clustering with deprivation and with the Peabody picture vocabulary. Oops. And then apparently I'm not describing it now, but we also observed that these variables, these sort of chaos variables, seemed like they were clustering with threats. So that's sort of variables that describe kind of unreliable interactions with the caregivers, and those in this analysis are all clustered with threat. So the network analysis confirms there's no single cumulative risk cluster. The deprivation variables do appear to cluster with cognitive outcomes, and the threat variables cluster with emotion or symptoms of psychopathology in early childhood. And observed neglect now clusters with deprivation. So in conclusion, across a bunch of different ways of testing this model so far, it really does look like deprivation and threat differentially predict child development outcomes. And that's using both data-driven and a priori approaches, and that they do lead to risk for psychopathology later. So these changes in cognition and the brain are the mediators of future outcomes in terms of psychopathology later. So this initial support for our model has been exciting. We're hoping in future work to test it a little bit more thoroughly. So I've sort of described some studies that are kind of hacked together based from various different data sources, and some work we were able to do prior to grant funding. And now we've got about, depending on how you count them, 3 different grants that are testing these ideas in larger samples with more robust measurement of deprivation, threat, our mediators, and our psychopathology outcomes. Oh, see, I thought that there was that there. So this is the final piece that we did, was we put socioeconomic status back into this analysis, and we showed that it ends up clustering with threat, but it is sitting right in the middle of the 2 clusters and is the source of information sort of going between the 2 clusters. So it's an important clustering variable. And I just wanted to say thank you to everybody in my lab, and thank you guys for your attention.
Thank you so much, Dr. Sheridan. What a rich talk. I took copious notes and I have a bunch of questions, but Steve Hinshaw beat me to the chase here. So Steve, go ahead, ask your question.
Hey, Margaret, wish you could be here
Steve.
live. And I just want to point out to the assembled audience that Margaret at Cal some years back essentially got a PhD in clinical science, clinical psychology in my lab, and a PhD in neuroscience in Mark Desposito's lab. And I think you can see the results of such a variegated cross-disciplinary education. So thanks, Margaret, for a great talk. So my question, I've got a lot of questions, but I'll focus on one. I just finished a review for annual research review of Journal of Child Psychology and Psychiatry on sex differences in ADHD and externalizing disorder, et cetera, et cetera. Could you comment on sex as a moderator of any of the findings you have here? You went really quickly, didn't have much time to point it out. Are you finding girls versus boys more susceptible to threat or deprivation on any of the dimensions, or in terms of any of the outcomes you're looking at?
Yeah, that's a really good question. We have looked a little bit at that in our smaller samples. I will say the sample where we haven't looked at that in is the fragile family sample, which is the only one we're likely to be powered to really see anything in. Yeah. So point taken, we should go do that. So I don't know the answer. In the small samples, we haven't seen any interactions with sex. Okay. And I think there are studies that exist out there that have examined exposure to violence in girls and how that might be related differentially to amygdala activation and development. And so I think there's pretty good reasons to think that you might see sex differences. We kind of haven't been powered. We haven't seen anything where we found it. And I think initially I was spending a lot of time trying to kind of hammer out the global idea, but I think it's probably about time to start thinking about that. So,
and just quickly as a follow-up, girls' brains develop earlier than boys overall. Might they be less susceptible to deprivation exposure than boys, or what do you think?
It's an interesting idea. I think it depends a little bit how that stuff gets embedded. I'm trying to think in the a lot of the work on deprivation, I tend to first think about findings in the Bucharest Early Intervention Project, because that's an example of institutionalization. This is a sort of kids in Romania who are randomized out of the institution. And the only interaction with gender that I know about there is that in adolescence, boys who were in the institutionalized group compared to the foster care group, so that's the random assignment, are more at risk for externalizing psychopathology, and specifically sort of conduct disorder type stuff. So I think it's either conduct disorder or callous unemotional traits, but it does look like you see like a differential prediction to psychopathology, but we haven't seen anything with the brain there. And I have looked. It's a really small sample though, so I don't expect to be able to really power to see anything.
Thanks a million. Great talk.
Thank you.
Alyssa Hamlet, go for it. Go ask your question if you're still with us.
Yeah, sorry. I just had a question, I mean, to follow up with Steve's, in that we know that early adversity and threat, I know Kate McLaughlin has found this, is associated with early puberty, and especially as girls are maturing earlier. I'm just curious if there's been any work done on how this might play out in terms of brain development, because it looks like some of the networks might be coming online earlier, or at least having more adult patterns, it appears. Anyway, anything about that would be interesting to hear about.
You may have heard this news already, so this might be what you're referring to, but Kate's got a great meta-analysis that she did with Natalie Kulish, who was one of her students, showing that threat but not deprivation is associated with that accelerated pubertal development. And that theory is sort of a theory that Jay Belsky and Bruce Ellis have talked a lot about, which is that adversity in general is going to kind of accelerate your development. And they really talk about it very broadly, sort of just unpredictable environments or dangerous environments and there are a lot of indicators of what might be a dangerous environment. But in her meta-analysis, it really looked like threat specifically was linked with that, but not deprivation. And I think that that is interesting and kind of consistent with this idea that there might be a more fine-grained way of thinking about adversity exposure, where threat really is a signal early in childhood that your environment is going to be dangerous, and deprivation is a different kind of signal. So it's not really signaling the danger of the environment so much as the sort of lack of necessity for complex dendritic arbors to solve complex problems later, for lack of a better way to describe that. Is that
yeah, that's well, just curious though, is that I am familiar with some of that lit, but do we see this play out? I mean, are there any studies looking at this in the brain as well? So I imagine that these changes occurring when somebody is maybe going through the pubertal process, when we get kind of the synaptic pruning already going on, like I don't know, is there any kind of interaction that might be, especially a risky time, especially if kids are maturing earlier than normative?
Yeah. I don't know of anything. The thing I'm thinking of is that, I don't know if you're familiar with Nim Tottenham's work about the sort of accelerated development of medial prefrontal amygdala connectivity, that you sort of, her hypothesis that you see that come online earlier in development in kids exposed to adversity, which has got kind of a broad definition of adversity, but I think that would be consistent with what you're describing. But other than that, I don't know of very many people who've done that. And there just aren't a lot of really big data sets with a lot of adversity and a lot of brain data. So there's sort of lots of adversity and not lots of brain and not lots of adversity, but the combination of that is pretty uncommon.
Very cool. Sorry, what was the last name of that person with the connectivity?
Tottenham.
Okay, great. I don't know that, so I will look that up. Thank you.
She's your woman.
So, Tony Yang, do you want to ask your question?
Sure. Thanks for an excellent talk. I was wondering if you could go into a little bit more depth about the relationship that you found that seems a bit more complex regarding cortical thickness and deprivation. So it was quite a tantalizing data that you showed, and I was just curious to know a little bit more about that relationship.
Totally. I'll tell you all of the dark and dirty secrets. So they're not that dark and dirty, but they're an interesting scientific adventure. So when we began our hypothesis, we really looked at some of the early literature on sensory deprivation, and that literature seemed pretty clear. There wasn't a lot of it, but it seemed like, when Wiesel and Hubel covered up the eyes of the cats, they saw thinner cortex, and it looked like then some subsequent follow-up work showed that that was because there was like decreases in dendritic arbors. And Marion Diamond, who's here, who taught me neuroanatomy, which is one of the things I am so proud of. She basically showed that if you take rodents of any age and you stick them into an environment that's lacking richness, you see this decrease in dendritic arbors as a result. And so I really thought this is kind of clear and we should see cortical thinning. And then the initial work in institutionalization was consistent with that. We saw that kids who've been exposed to institutionalization in cortical thickness kind of all over their cortex. And it seemed like a pretty cut and dry guess about what was going to happen, or I think we call those hypotheses in science. And then this future work has really refuted that. So it's been more complicated. And when we went and looked in little kids there, we saw that they had thicker cortex. It was just in association with deprivation. It was robust to controls for threat. Threat wasn't associated, but it was thicker. And then more recently, I'm writing up a paper right now in the Bucharest Early Intervention Project, where in adolescents, we also see that they have thicker cortex. And so I think I'm left a little bit trying to figure out what's going on. I went back to the animal literature, and since the time we first were reading that, there is a lot of interesting work that shows that there's sort of two different things that can happen. One is that the sensory deprivation sort of teaches the brain that there is no input coming and you prune away what you don't need. The other possibility is that you might put the brain into a state of sort of quiescence, so it's waiting for input, and in that case you'd expect it to be thicker for longer. So in a case where you're getting kind of like intermittent, not like complete lack of sensory input, but some sensory input, maybe that's what you're going to see. And so my only guess is that this is the thing that we're thinking about with association cortex is more like that, and that actually we should expect thicker cortex. But I wish I had sort of like a formal good answer, but I just have more information that the direction isn't 100% clear to me right now, although it's a very consistent association with cortical thickness.
All right, thank you very much. I just wanted to add in Marion Diamond. When I was in high school at El Cerrito High School, I didn't realize as a high school student, since my prefrontal cortex wasn't fully myelinated, that I shouldn't email and call and contact Marion Diamond to ask her to give a talk at my high school. So I went ahead and contacted her, and she actually replied and came over and gave my high school class a really great talk on neuroscience, which really got me interested in the whole field. So I just wanted to.
If we could, she is somebody we should all aspire to be, I feel.
And Marion passed away a couple of years ago, Margaret, if you didn't know. So,
I didn't know that.
But you have to look at the PBS special, My Love Affair with the Brain, which is a documentary on Marion, which is incredible. So,
Andrew Fox, you had a question. You're raising your hand.
I was curious about whether you've, examined so far or just would have any hypotheses based on DMAP around exposure to like racial trauma, or especially like in the conceptualization of like, well, some forms of racism may be more overt and threatening, but some may be more in the deprivation of resources to communities. And just wanted to hear your thoughts about that.
Yeah. I definitely am super interested in that. And I have, in our more recent studies, we've started measuring both microaggressions and also sort of racial stress on a couple of different scales. And I think the thing that you said basically, which is that I think that that is almost certainly a source of threat, which is unmeasured in a lot of studies, that there's sort of like the potential for violence that exists and then potential experiences of violence that people have had, that form of discrimination and racism is probably a form of threat exposure. And I'm hoping that we've measured some of that in these more recent studies. And then I think that the structural racism, I think that's kind of obvious. Like if you're reducing resources as we are to a whole group of people, that it's going to increase deprivation exposure for those groups.
Thank you.
Margaret, thank you, Andrew, for that. On that note, I'm wondering how your work informs social policy and public policy, right? You wrote an op-ed piece in the New York Times about separation of children at the border from their caregivers and how that may impact their trajectories. And so on that, I started thinking about government assisted allowances for families living in poverty, and well, how do you think about that? How do you think about your work impacting policy?
I think a couple things. One is that there's sort of a way I think it might impact clinical work, and then there's a way that I think it might impact policy, which are different. I think in the case of policy, the more evidence that I have that is experimental or association evidence that those kinds of deprivation exposures are shaping brain development in kids, I hope that that is more and more robust evidence that we have to be thoughtful about the kinds of experiences that kids are having. And it doesn't matter if we mean to do well by them, if we are instead hurting them, that is fundamentally we're shaping their brain, and that those the way that we shape their brain will live with them for the rest of their life. And it's not like there's no source of remediation. Certainly people are incredible, but that it still means that there has been an impact early on. And in the case of that op-ed, one of the things I talk about when I've talked about that idea, I was really there talking a lot about the Bucharest Early Intervention Project and the experimental evidence that early deprivation really shapes brain development. And I went back and looked at the kinds of things we do in rodents, and I saw that the sorts of exposures in rodents, the duration of exposure to adversity is about like the same amount of their life. So it's like 3 or 4 hours of the pup's life. That same amount, the percentage of their life, their early life with just exposure, is the same thing as the 3 months that on average kids who were put separated from their parents were exposed to. So it's the same percentage of time. So if we want to think about whether or not that was an impactful exposure, the evidence that we have so far is that it certainly should be, like that separation from their parents should have had a profound effect on them if they were sort of kept in an institutionalized state that didn't involve caregiver interactions. So I think that in one way, I kind of just hope that I'm fomenting, I think we all are who are advocating for children, just keep trying to foment concern and hope that that changes into policy. I think there is less, the way that I think that there's sort of clinical or sort of differential reasons to think about deprivation and threat with regards to policy or clinical intervention is that what we think is going on with kids. So I think that sometimes we're faced with things like big differences in kind of school performance or kinds of different outcomes having to do with school, basically. And we think of them as being related to stress. And when we do that, we do sort of stress-based interventions. So we think about doing mindfulness, and we think about doing sort of like interventions that are going to support kids emotionally. I think that that is great. But I think that if what they're lacking is scaffolded cognitive interactions, and we need to also address that. And in some ways, that's become kind of an old idea in psychology. There was sort of like fragile families study started a long time ago, and people said, oh, okay, sure, cognition, but like, let's talk about stress. And I think that I'm trying to just say, let's keep talking about both of them because they might be really important in different ways. And if you want to affect school outcomes, you might need to do one thing. And if you want to affect emotional health, you might need to do something else.
Yeah, you're not going to have an effective clinical session when your child in front of you is hungry.
Right, exactly. Gotta do both things. That's right.
Yeah. Thank you so much, Margaret.
Thank you.
And thank you for staying over with us and the folks that did. And I appreciate it so much. Can't wait to see you in person.
Fun. Yeah, you too. It was so fun to see so many friendly faces. It was great to see everybody.
Take good care. Bye.
Bye.